Welcome to the Deep Dive. Today we're plunging into a world that is, well, mostly invisible, yet it pulls the strings on almost everything in your life. We're talking about microbes, these tiny living things. They shape everything, the air we breathe, the food we eat, even our own health.
It's quite a paradox, isn't it? Invisible, yet completely fundamental.
Okay, let's unpack this. This deep dive is going to reveal how these tiny organisms have shaped our world and continue to do so.
Yeah.
And we're using a really solid source, microbiology. An introduction, 13th edition.
Right.
Our mission for you, the listener, is to get a rapid but comprehensive understanding of this vital field.
And what's fascinating here is just how profoundly our understanding of these minute living things has evolved. I mean, it's led to revolutions in medicine, agriculture, environmental science.
It really shows us this unseen majority, doesn't it?
Exactly, an unseen majority that dictates so much of what happens on Earth.
And it really is an unseen majority. You know, when most of us hear microbe, we immediately think disease,
Right.
germs.
That's the common perception, yes.
But the surprising truth is, the vast majority of microorganisms are actually essential. They form the fundamental balance of life, and they are quite literally everywhere.
Indeed. Just consider your own body for a moment. An adult human has about 30 trillion body cells.
Okay.
But you actually harbor around 40 trillion bacterial cells. This whole collection, we call it your normal microbiota or your human microbiome.
40 trillion, that's truly mind-blowing.
It is.
So the real insight here isn't just that microbes are inside us, but that our very biology, our digestion, our immune system is fundamentally built on this partnership.
Precisely. They aren't just freeloaders. For instance, bacteria in our intestines, like some strains of E. coli, help us digest food.
Got it.
And they synthesize essential vitamins. Think vitamin B for metabolism, vitamin K for blood clotting.
Right.
Plus, they act as, well, biological bouncers. They prevent harmful species from getting a foothold. And they even help train our immune system from an early age.
So we start getting them right from birth.
Exactly. We begin acquiring these microbial partners from the moment we enter the world. Understanding this intricate relationship is so critical, actually, that major initiatives like the Human Microbiome Project and the National Microbiome Initiative were launched.
Ah, right. I've heard of those. To map everything out.
To map it out, yeah, and to expand our understanding of just how microbes affect human health, but also other ecosystems, too.
Because their importance extends far beyond our bodies, doesn't it? Environmentally.
Oh, absolutely. Microbes are the base of food chains in oceans and lakes. They're the planet's great recyclers.
How so?
They constantly decompose waste, dead organisms, recycling vital chemical elements like carbon, nitrogen, oxygen, sulfur, phosphorus, turning them back into forms that plants and animals can actually use.
It's incredible.
And get this, only bacteria can naturally convert atmospheric nitrogen into a usable form. That's crucial for all life. Plus, many are key for photosynthesis, producing food and oxygen.
Wow. Okay, so they run the planet's chemistry, basically.
In many ways, yes. And their utility isn't just limited to natural processes either. Microorganisms have immense commercial and industrial applications.
I imagine so.
Yeah.
Like producing chemicals.
Yeah.
Vitamins, enzymes.
Exactly. Vitamins like B2, B12, organic acids, enzymes, alcohols. There's a fascinating historical example, actually.
Oh, yeah.
During World War I, Chaim Weizmann discovered how certain microbes produce acetone and butanol.
Okay.
That was absolutely critical for making smokeless gunpowder. It significantly impacted the war effort.
No kidding, just from microbes.
And beyond chemicals, think about the food industry. Vinegar, sauerkraut, pickles, soy sauce.
Cheese, yogurt, bread, beer and wine.
All of those. Microbes are the silent partners in their creation.
And now with biotechnology, we're even engineering microbes to produce things they normally wouldn't, right? Like human insulin or proteins for vaccines.
That's right. It's a huge field.
So, okay. These tiny powerhouses are essential. They're everywhere. They're even inside us. But with such incredible diversity, how do scientists even begin to categorize this microscopic world? It seems overwhelming.
Well, that's where the system of scientific nomenclature comes in. It provides order, established by Carolus Linnaeus way back in 1735.
Linnaeus, right? Binomial nomenclature.
Exactly. It assigns each organism two names. Think of it like a first and last name. The first is the genus, which is always capitalized,
Yep.
followed by the specific epithet or the species name, which is not capitalized. And both names are either italicized or underlined.
Right, so you get names like Staphylococcus aureus, And those names often tell you something, don't they?
They often do if you break that one down. Staphylo describes their clustered arrangement like grapes. Coccus means they're spherical cells. And aureus is Latin for golden, which refers to the color of their colonies on agar plates.
Descriptive. What about Escherichia coli?
Named after the physician Theodor Escherich, who discovered it. And coli indicates its habitat, the colon, or large intestine.
It's a very neat, descriptive, and importantly, universal system. Everyone uses it.
Indeed. And within that system, we differentiate microbes based on their structure and function. Let's maybe start with bacteria.
Okay.
These are relatively simple, single-celled organisms. We classify them as prokaryotes.
Meaning no nucleus, right? Their DNA is just sort of there.
Correct. Their genetic material isn't enclosed in a membrane-bound nucleus. They typically have cell walls made of a substance called peptidoglycan.
Peptidoglycan.
They reproduce usually by just splitting in two. That's binary fission. And they come in various shapes. Rod-like ones called bacillus, spherical ones called coccus, or spiral shapes.
Got it. Then there are archaea. They sound similar.
They are similar in that they're also prokaryotic, single-celled. But crucially, their cell walls lack peptidoglycan, and their genetics are actually quite different.
And they like extreme places.
That's their claim to fame, often. Found in extreme environments, methanogens produce methane gas. Extreme halophiles love super salty places. Extreme thermophiles thrive in intense heat.
Wow. But they don't make us sick.
Importantly, no known archaea cause human disease, which is interesting.
Okay. Moving on from prokaryotes, eukaryotes. They have a nucleus.
Right. Eukaryotes have a true nucleus enclosing their DNA, and this group includes fungi.
Like yeasts and molds.
Exactly. Fungi can be unicellular, like yeasts, or multicellular, like molds and mushrooms. Their cell walls are primarily made of chitin.
Not cellulose like plants?
Nope, chitin. And unlike plants, they don't photosynthesize. They get nutrients by absorbing organic material from their environment, soil, water, even animal or plant hosts. Molds often form visible, cottony masses called mycelia.
Okay, fungi. What else in eukaryote?
Protozoa. These are unicellular eukaryotes. They're incredibly diverse, especially in how they move. Some use pseudopods, sort of like temporary false feet.
Like amoebas?
Like amoebas, yes. Others use flagella, which are long whip-like tails, or cilia, which are short hair-like projections. Some are free-living, others are parasites.
Gotcha. And algae?
Algae are photosynthetic eukaryotes. They can be unicellular or multicellular. Many have cellulose cell walls, like plants.
So they make oxygen.
Yes, they're abundant in water and soil and play a crucial role producing oxygen and carbohydrates through photosynthesis.
Okay. Now, viruses. They always seem like the odd ones out.
They really are. Viruses stand apart because they are acellular. They aren't cells at all.
Not alive or...
It's debated. They're incredibly small, usually need an electron microscope to see. A virus particle is basically just a core of genetic material, either DNA or RNA, never both surrounded by a protein coat. Sometimes there's an outer lipid envelope, too.
And they need us or other cells to reproduce.
Exactly. They can only reproduce by hijacking the machinery of a living host cell. They're essentially obligate intracellular parasites. Outside a living host, they're inert, just particles.
Fascinatingly simple, yet complex.
And finally, while not strictly microorganisms, we should mention multicellular animal parasites.
Like worms.
Yes, flatworms and roundworms, collectively called helminths. They aren't microbes themselves in their adult forms, but their microscopic eggs and larvae are often identified in labs using similar techniques to microbes, so they're studied within microbiology.
Right, so bacteria, archaea, fungi, protozoa, algae, viruses, and even some parasites. It's a huge range.
It is, and this entire classification, particularly Carl Woese's three-domain system from 1978, which groups all life into bacteria, archaea, and eukarya, isn't just academic labeling.
It's more fundamental.
It's our roadmap. It helps us understand the deep evolutionary connections across all life, which is crucial for studying their roles and relationships.
This order we have now, this system, it's a far cry from earlier times, right?
Yeah.
When the very existence of microbes was unknown.
Oh, absolutely. It really started with the first observations. Think back to 1665, Robert Hooke peers through his microscope at Cork.
And sees cells.
Sees little boxes, which he called cells. His discovery was the beginning of the cell theory, the revolutionary idea that all living things are composed of cells.
But he didn't see bacteria, right? His microscope wasn't strong enough.
It seems not clearly enough. It was actually Anton van Leeuwenhoek, a Dutch merchant, who between 1673 and 1723 was likely the first person to observe live microorganisms.
With his own homemade microscopes?
Yes, significantly better ones. He meticulously drew these tiny animalcules, as he called them, from rainwater, teeth scrapings, all sorts of things. He really opened up the microscopic world.
But once this invisible world was glimpsed, the big question became, where did these tiny things come from?
Exactly. And for centuries, many believed in spontaneous generation.
Life from non-life, like flies from manure or maggots magically appearing on meat.
That was the common belief, that life could just arise spontaneously from non-living matter.
But people started questioning that.
Yes. Francesco Redi, an Italian physician, challenged it back in 1668. He did experiments with meat in jars, some open, some sealed, some covered with netting.
And showed maggots only appeared when flies could lay eggs on the meat.
Precisely. He disproved it for maggots.
Yeah.
But the debate raged on for microorganisms. John Needham in the mid-1700s seemed to support spontaneous generation. He heated broths, but they still became cloudy with microbes.
But then Spallanzani came along.
Yes, Lazzaro Spallanzani, about 20 years later, he repeated the broth experiments, but heated them after sealing the flasks.
And they stayed clear.
They stayed clear. He argued Needham's results were likely due to microbes getting in from the air after boiling, but before sealing. Still, some argued heating destroyed a vital force needed for spontaneous generation.
It took Pasteur to really settle it.
It did. Louis Pasteur in 1861. His famous S-shaped flask experiment was definitive.
Explain that again.
He boiled broth in flasks with long curved necks, S-shaped. Air could get in, but dust and microbes were trapped in the curve.
And the broth remained sterile.
Exactly, until he tilted the flask so broth touched the trapped dust. Then, microbes grew. It conclusively disproved spontaneous generation for microbes.
And that wasn't just academic, right? It led to practical things.
Absolutely. Pasteur's work laid the foundation for aseptic techniques procedures that prevent contamination by unwanted microbes. These are fundamental in labs and medicine today.
So Pasteur really kicked things off.
His work truly ignited what we now call the first golden age of microbiology, roughly 1857 to 1914. A period of rapid advances.
Like figuring out fermentation.
Yes. Pasteur discovered that yeasts ferment sugars to make alcohol, but he also found that different bacteria could convert that alcohol into vinegar, causing spoilage in wine and beer.
Which led to pasteurization.
Exactly. He developed pasteurization, a gentle heating process to kill most of the spoilage bacteria without ruining the beverage. Still used today for milk, juice, etc.
And connecting microbes to chemical changes like spoilage led to an even bigger idea.
The germ theory of disease. If microbes could cause chemical changes, maybe they could also cause disease in plants and animals, including humans.
That must have been revolutionary. People thought disease was caused by, what, bad air, curses?
Misdeeds, imbalances in body humors, miasmas, or bad air, yes. The idea that tiny, invisible organisms were the culprits was a massive shift.
Did Pasteur prove it?
Well, others built on his work. Agostino Bassi showed a fungus caused silkworm disease back in 1835. Pasteur himself later identified a protozoan causing another silkworm disease.
And this had practical results in medicine, too, like surgery.
Definitely. In the 1860s, Joseph Lister, an English surgeon, applied the germ theory directly. Inspired by Pasteur, he started treating surgical wounds with phenol, a disinfectant.
And infection rates plummeted.
Dramatically. He proved microorganisms cause surgical wound infections, leading to aseptic surgery techniques.
But the first direct proof that a specific bacterium caused a specific disease.
That's Koch.
Robert Koch, a German physician. In 1876, he proved that the bacterium Bacillus anthracis caused anthrax. Anthrax, that terrible disease in cattle and sheep, sometimes humans.
Yes, and Koch didn't just find the bacterium. He established a sequence of experimental steps to rigorously prove the link. We now call these Koch's postulates.
Still used today.
They remain invaluable for identifying the causative agents of infectious diseases. You have to isolate the microbe from a diseased animal, grow it in pure culture, infect a healthy animal with it, see the same disease, and then re-isolate the same microbe.
Wow, very methodical.
Extremely. This golden age wasn't just about understanding cause, but also prevention. Vaccination started even earlier, actually.
Jenner and smallpox.
Edward Jenner, 1798. He observed that milkmaids who got mild cowpox didn't get deadly smallpox, so he deliberately inoculated a boy with cowpox pus.
Risky?
Very. But it worked. The boy became immune to smallpox. Years later, Pasteur built on this, developing vaccines using weakened or virulent microbial strains. He coined the term vaccine from vacca, Latin for cow, honoring Jenner.
Okay, here's where it gets really interesting for me. These foundational discoveries weren't just about understanding. They were about intervention, setting the stage for truly fighting disease.
Absolutely. And that intervention really blossomed into what many call the second golden age of microbiology, starting around the 1940s. The main focus became chemotherapy.
Using chemicals to treat diseases.
Exactly. The dream was finding a magic bullet, a drug that could kill the pathogen without harming the patient. Paul Ehrlich was an early pioneer.
Right, you mentioned him. Selversen for syphilis.
Yes, in 1910. Selversen, an arsenic derivative. It was one of the first synthetic drugs successfully used in chemotherapy. A huge step.
But the game changer was antibiotics.
Undoubtedly. And it started famously with an accident. Alexander Fleming in 1928.
The Mulvey Petri dish.
That's the one. He noticed a clear area around a colony of penicillium mold where bacteria weren't growing. He isolated the active compound penicillin, the first true antibiotic.
And it revolutionized medicine, saved countless lives in World War II and beyond.
It truly did. But as powerful as antibiotics were and are, they brought a new, very serious challenge: resistance.
Right. The microbes fight back.
In a way, yes, it's evolution in action. Through random genetic changes, some microbes develop traits that let them tolerate drugs that would normally kill them or inhibit their growth.
How do they do that?
Various ways. Some produce enzymes that break down the antibiotic, others change their surface structures so the drug can't attach properly, or they develop pumps to eject the antibiotic if it gets inside.
And when we use antibiotics, we kill the susceptible ones, leaving the resistant ones to multiply.
Precisely. Which leads to infections that are incredibly difficult, sometimes impossible to treat. Think of MRSA methicillin-resistant Staphylococcus aureus, or VRSA vancomycin-resistant, or multi-drug-resistant tuberculosis, MDR-TB. It's a massive global health crisis.
Scary stuff. So these discoveries, both the good and the bad, they spurred specialization within microbiology, right?
Yes, the field branched out considerably. Bacteriology, the study of bacteria, continues to uncover new things, like finding Theomargarita namibiensis in 1997, a bacterium so large you can see it without a microscope.
Wow. And mycology. Fungi.
Study of fungi, yes. It's gained importance partly because fungal infection rates seem to be rising. Things like valley fever, Coccidioides immitis, infections in California, possibly linked to climate change.
Parasitology, too. Protozoa and worms.
Yes, studying parasites like the guinea worm, which fascinatingly might have inspired the rod of Asclepius, that ancient symbol of medicine with the snake wrapped around a staff.
Huh. And immunology. The study of our immune system.
A huge field. Vaccines are a cornerstone, obviously. Smallpox was eradicated globally thanks to vaccination. We discovered interferons, substances cells produce to fight viruses, but challenges remain, like developing an effective AIDS vaccine.
Virology viruses must have exploded once we could actually see them.
Absolutely. It started with Dmitry Aronofsky realizing the cause of tobacco mosaic disease could pass through filters that blocked bacteria. Then Wendell Stanley crystallized the virus, showing it had properties of chemicals and living things. The electron microscope revolutionized the field.
And underlying all of this, genetics, especially molecular genetics.
Yes. So much of our fundamental understanding of how genes work, DNA structure, gene regulation, how genes code for enzymes came from studying microbes first.
Why microbes?
They're ideal models. They're less complex genetically than plants or animals, and they have very short life cycles. You can observe generations and genetic changes much faster. Think Beadle and Tatum connecting genes to enzymes, Avery, McLeod, and McCarty proving DNA carries genetic information, Watson and Crick figuring out DNA structure. Much of it involved or was influenced by microbial studies.
Okay. This raises an important question then. As our knowledge is exploding like this, what new frontiers emerged? And what unexpected challenges did these breakthroughs bring along?
That brings us more or less to the present day, what some call the age of bacteria, or maybe the third golden age. It's really driven by new technologies, especially in DNA sequencing and computing power.
Genomics. Studying the entire genetic makeup.
Exactly. Genomics allows us to study all of an organism's genes. This is huge because we can now classify and understand microbes that we can't even grow in the lab, and there are vast numbers of those. It's revealing incredible diversity we never knew existed.
And that leads to things like recombinant DNA technology.
Directly. Paul Berg's work in the late 1960s figuring out how to combine DNA from different sources like human or animal DNA with bacterial DNA was pivotal.
And now we use it all the time.
All the time. It allows us to harness microbes as tiny factories to produce important substances they wouldn't normally make: human insulin, vaccines, enzymes, hormones.
It's incredible. And gene therapy fits in here too, using viruses to deliver genes.
Yes, that's another application. Using harmless modified viruses to carry missing or defective genes into human cells. It holds promise for treating genetic disorders like severe combined immunodeficiency, SCID, Duchenne's muscular dystrophy, cystic fibrosis, though it's still complex and faces challenges.
But the potential is huge.
Immense. And these genetic technologies are also revolutionizing agriculture, genetically altered bacteria protecting crops from frost or insects, for example.
Okay, so amazing progress. But despite all this, microbes still pose huge challenges, especially with human disease.
They absolutely do. Our body's resistance, its natural ability to ward off disease, is constantly being tested by the disease-producing capabilities of microbes. And one really significant factor in modern infections is biofilms.
Biofilms, you mentioned those slimy layers.
Exactly. Complex communities of microbes sticking together, usually encased in a gooey matrix they secrete. Think plaque on your teeth, slime on river rocks.
They sound bad.
They can be a double-edged sword.
No.
Sometimes they're beneficial, like protecting our mucous membranes, but often they're problematic. They can clog water pipes, industrial equipment.
And medical devices.
Critically, yes. They readily form on things like joint prostheses, heart valves, catheters. These biofilm infections are notoriously difficult to treat.
Why?
Because the bacteria huddled together inside that slimy matrix are often protected. Antibiotics have a hard time penetrating, and the bacteria may be in a less active state, making them less susceptible. It's a major source of persistent and recurring infections.
Okay, biofilms. And then there are emerging infectious diseases, EIDs.
Right, EIDs. These are diseases that are new or were known but are rapidly increasing in incidence or geographic range.
What causes them to emerge?
A variety of factors. Evolutionary changes in existing microbes, making them more dangerous or able to infect new hosts. The spread of microbes to new areas due to modern travel and trade, increased human exposure resulting from ecological changes like deforestation or climate change. Sometimes pathogens jump from animals to humans.
We've certainly seen plenty of examples recently. Zika?
Zika virus disease, yeah. Spread by Aedes mosquitoes, also sexually transmitted, first really noticed globally around 2015. Often mild symptoms in adults, but devastating birth defects if a pregnant woman is infected.
And flu viruses are always changing.
Constantly. We had the H1N1 swine flu pandemic in 2009, and there's ongoing concern about avian influenza strains like H5N1 or H7N9 potentially adapting to spread easily among humans.
MERS, too, that coronavirus.
Middle East respiratory syndrome, MERS-CoV, emerged around 2014, another coronavirus related to SARS with a high mortality rate linked primarily to the Middle East.
And Ebola, Marburg, those hemorrhagic fevers.
Ebola virus disease, known since the 70s, but caused a massive epidemic in West Africa in 2014. Marburg is similar, also causing hemorrhagic fever. Both are incredibly dangerous, spread through contact with body fluids.
And underlying all this, the antibiotic resistance crisis just keeps getting worse.
It really does. We talked about Staphylococcus aureus progressing from penicillin-resistant to MRSA, then Visa, then VRSA. Multi-drug resistant TB is a huge problem globally, and strains like the epidemic Clostridium difficile that emerged in 2004, often in hospitals, cause severe diarrhea and are hard to treat because of resistance.
It makes you think twice about using antibacterial soap for everything.
Exactly. Routine use of antibacterial soaps can contribute to resistance. Plain soap and water are usually sufficient for hand washing. We need to preserve the effectiveness of the antibiotics we have left.
If we connect this all back to the bigger picture, these emerging diseases, the resistance problem, they really highlight how dynamic microbes are. They're constantly evolving, interacting with us and the environment. It's a continuous challenge.
It truly is a continuous challenge to understand them, to manage them, and to adapt ourselves.
So what does this all mean? I mean, we've gone from not even knowing microbes existed to understanding their genes, but they still surprise us.
It means that microorganisms, from their absolutely essential roles in keeping the planet running and keeping us healthy, to their capacity to cause devastating diseases, are a fundamental, inescapable part of life on Earth.
Yeah.
Our deep dive today, drawing from that foundational knowledge in microbiology, it really reveals a field that's always discovering. Every new insight seems to lead to new challenges, but also new solutions and new questions.
So here's something to think about. What new beneficial roles for microbes are still out there, waiting to be discovered? And conversely, what new microbial challenges might emerge from that invisible world that, as we've seen, continues to shape our very existence?
It's a constant frontier. Thank you for joining us on this deep dive into the fascinating world of microbiology. We hope you feel a little more well-informed, maybe even amazed, by these incredible tiny forces that shape our world.
Thanks, everyone.